Perfect. Thank you for that introduction. As you said, my name is Nicole Xu. I'm a graduate student at Stanford working with Professor John DeBerry at Caltech. And today I'll be talking about using biohybrid robotic jellyfish for future applications in ocean monitoring. So to motivate this work more broadly, the ocean makes up about 3/4 of the Earth's surface, but more than 80% of the ocean is unmapped, unobserved, and unexplored. However, the ocean is responsible for processes such as oxygen production, thermal regulation, and hosting diverse ecosystems. So it's important to monitor the ocean to track changes in temperature, acidity, and concentrations of nutrients in order to detect and prevent situations like coral bleaching, as shown here in an example of a healthy versus an unhealthy coral. As another example, we can look at algal blooms like the ones in Lake Erie caused by pollution. But in addition to this, we also want to explore the ocean for the sake of discovery, to find more creatures like this really cute sea bunny nudibranch, and to understand complex underwater ecosystems better. So our current methods of ocean monitoring include using ROVs and AUVs. Remotely operated vehicles, ROVs, are kind of like small cars that are attached to boats via these long cables. On the right is a Slocum glider, which is an example of an AUV, an autonomous underwater vehicle. And these are both useful for ocean measurements because they can reach areas that boats and divers traditionally can't. But there are also some challenges. So they can be really costly in terms of manpower as well as manufacturing. They can also be very bulky, as shown here, and they can consume a lot of power. In addition, they can leave these unnatural wakes, like behind these boats and wakeboarders here as an example. And these wakes can potentially disrupt more sensitive parts of the ocean. The question is, how can we expand upon these existing technologies to monitor the ocean? Our goal is to use technology to collect data about ocean environments. So to do this, we need to be able to explore new areas of the ocean that might be farther away or harder to reach. So we need technologies that are low power, low cost, and minimize disturbances to aquatic life. We can achieve this by combining different technological advances as well as experimental work and computational tools. And I'll be stepping through how my PhD work has incorporated all three of these with an emphasis on the technology and the experimental side in my research and where we're heading in terms of incorporating more HPC and AI into this work. So to address these needs in ocean monitoring, we can look to nature as an inspiration. By using robots that swim like natural animals, we can create more energy efficient aquatic vehicles that leave more natural wakes to minimize these disturbances. And they can be used to collect images, take data from the environment, and even take samples of water from new locations that we haven't reached before. So as a few examples, here's a robotic fish, a robotic manta ray, and jellyfish. So robots like these can be used in the future, but the question is, what animal model do we choose? And of course, I'll be talking about jellyfish. So we want to use jellyfish as a compelling model organism because despite 500 million years of evolutionary pressure, their body structure has remained largely unchanged. They're also found in a wide range of environments, from extreme salinities to deep depths thousands of meters below the surface. So what about their simple body structure is so advantageous? It's been hypothesized that their body conservation is largely because of energy efficiency. So to look at that, we can use one metric of energy efficiency, which is the cost of transport, or the energy per mass per distance traveled. You can think of it as the opposite of miles per gallon. So a low cost of transport is more energy efficient. So when we're comparing different costs of transport, we're looking for the lowest value. The plot shows the cost of transport on the y-axis and the mass of animals on the x-axis. And it's important to note that these are log scales. So as shown for different types of locomotion, for animals of the same mass, we can see that running is the least energy efficient, followed by flying, then different types of swimming, with jellyfish swimming all the way at the bottom. So from observations of natural animal behavior, we can conclude that jellyfish are energy efficient. But folding it back into the discussion about bioinspired robots. If we want to create a jellyfish robot, then you can imagine that there's this spectrum of constructs that we can choose from. So on one end of the spectrum is a purely robotic construct like RoboJelly, which is great because we can control all of the design parameters, all of the inputs and outputs. But the main limitation here is high power consumption. So a lot of these entirely mechanical robots still tethered to some external power source. On the opposite end of the spectrum is more of a biological construct. So this is an artificial jellyfish called the medusoid, made from seeding rat heart cells onto a silicon scaffold. And while this is a great proof of concept, these constructs can only survive in very specific environments in the lab. So they can be limited in terms of applications for ocean monitoring. So what is our approach, and then where do we fall along this line? Well, to jump to the end of the story, our approach is to use the animal itself as a natural scaffold. So the idea is we can embed a microelectronic swim controller into a live animal to create a biohybrid robot. And the end goal is to send these biohybrid robotic jellyfish out into the ocean to collect data. We've started some preliminary experiments to get the feasibility of that in the ocean, but to get there, I'm going to tell you the story of building this biohybrid robotic jellyfish and what we've tested and discovered so far. So to give you a roadmap, I'll start by explaining how jellyfish naturally swim, how we can control their swimming muscles using electrodes, the process of miniaturizing this system into a portable swim controller, and what scientific questions we can answer with this biohybrid robotic system. Then I'll finish by talking about the fieldwork in the Atlantic Ocean, which is ongoing work, and future directions for this robot and incorporating more HPC and AI. So before we can get there, let's first discuss how jellyfish naturally swim. This is a jellyfish oriented upside down. The underside is the subumbellar surface, and the top of the animal is the exumbellar surface. This is an image of a jellyfish placed in a dish, subumbellar surface facing up. Notice on the edge of the bell are these 8 little divots that each contain a pacemaker, or a sensory organ that innervates the muscle, which is this single monolayer ring on the subumbellar surface. So in order to swim, the jellyfish activates these pacemakers, which causes muscle contractions to begin at those specific sites. This is called the power stroke. There is also a recovery stroke in which the jellyfish muscle is passive and relaxed. And it's important to note that this is basically an all or nothing muscle activation. So even if only one or a few of these pacemakers fires, it will eventually cause the entire muscle to activate, starting from those few locations. But we're not limited to the biology here. So instead of relying on the animal's natural pacemakers to initiate muscle contractions, we can use electrodes to activate the muscles, starting from the locations where we actually place the electrodes. So the inputs are signal voltage, pulse width, the signal period, and where we place the electrodes on the bell. These are the characteristics to determine what signals actually incite muscle contractions in jellyfish. And today I'm going to be focusing on the frequency as well as the placement of the electrode on the bell. From these inputs, we can get outputs, which are muscle contractions or pulses. And to look at that, I'm going to use tag displacements, and I'll explain what that means on the next slide. Lined in yellow just to give a visual representation of where the animal is. And there are also these red tags implanted, and I'm going to circle them in blue. So if you pay attention to any one of these blue circles, you can see that every time the jellyfish contracts, which is somewhat irregular, you can see that the red dots displace slightly. So we can then get plots of the tag displacements over time. This is an example of one tag displacement for one jellyfish. And so this is one example of one jellyfish, but you can also see that if we stimulate the animal at 1/4 hertz, we can get a very regular pulse. So the black vertical lines are when we're externally stimulating the animal, and you can see that the animal follows very well. Occasionally, we can get double pulses or even triple pulses. So if you see at time around 12 seconds, you can see a very clear double pulse here. The animals can follow this frequency up to 1 hertz very well, but if we drive the animals much faster, so this is shown at 2 hertz, the animals are then unable to follow this frequency. So from this information, we can get a frequency spectrum. This is the single-sided amplitude spectrum over frequency for natural animal behavior. There is about frequency at about 0.16 hertz, with a spread of larger frequencies between 0.4 hertz and 1 hertz to account for multiple or successive pulses per bout. So we can compare the natural case to the stimulated case at 1 hertz, and you can see that there's a very sharp, steep peak at 1 hertz, which is the frequency of input. From this, we can look at the power spectral density. So again, this is an example at 1 hertz, and we can produce different power spectral density curves like this for a variety of different frequencies and plot them all together into this contour map, which shows the frequency output over the frequency input. Each PSD is a single vertical line here, shown in white, and the color is the amplitude of the PSD interpolated. I just want to point out a couple of features here. So the red diagonal line is a successful one-to-one response. The white dashed line is the maximum pulse frequency according to a proposed biological limit from the 40s. And the red dashed line is the peak frequency of unstimulated animals, so what the animals are naturally doing in the absence of external stimuli from us. The major takeaway from this plot is that we can reliably stimulate the muscle between 1/4 hertz and 1 hertz, and anything above 1 hertz basically defaults to frequency responses that are similar to unstimulated cases. So now that we know what signals we can use, we can investigate where to place the electrodes on the animal for successful stimulation. So I tested 4 different locations on the animal. One is at the stomach, one is midway between the stomach and the edge of the animal, one is at the edge on a pacemaker, and one is elsewhere on the margin of the animal. I just want to talk a little bit about future work and how we can incorporate different sensors like pH or pressure, which we can then put on the animals and send them out into the ocean in the future. And I'll highlight a little bit about that later as well. So when we look at the 4 different placements of the animal, we can see that these 4 plots are statistically insignificant, or they're statistically insignificant in terms of the differences. So we can successfully stimulate the animals at all 4 regions. So the conclusion is we can place electrodes anywhere for consistent muscle responses. We can also use models to map the trajectory of the animals and really use computational fluid dynamics to determine where we can place sensors optimally on the animal. So again, these are not electrodes, but other sensors like pressure or temperature or pH for untethered experiments in the ocean. So this is an example of a CFD jellyfish on the left and a real jellyfish on the right, and you can see that the stopping vortices are pretty similar. And if we look at a CFD model of the out-of-plane vorticity on the left and of vertical velocities on the right, what we can hope to do is eventually track the trajectories and be able to close the feedback loop. So we can use models in order to advance our knowledge and figure out how we can integrate these animals and send them out into the ocean consistently. So going back to building this biohybrid robotic jellyfish, the next thing I wanted to do was miniaturize the system. So in order to do that, we looked at a couple of different design choices. So harkening back to what we need our device to do, we wanted something that was very small, that was neutrally buoyant to salt water, and something that was low power and low cost. I'm going to point out a couple of features here. So this is a plastic housing with a wooden pin that inserts into the animal. And we tested a couple of different engineered materials before we discovered that wood was actually the best natural material to use because they have microscopic barbs that hook into the tissue very well. We also use a plastic cap just to waterproof all the electronics. And this microcontroller, a tiny lily, as well as a battery, and 2 electrodes with LEDs that flash every time that there's a signal that goes to the animal. So this is what the system looks like when we put it together. The microelectronics are within the plastic housing, and you have the 2 electrodes going into the side. This is what it looks like when the system is embedded into the animal itself. And I'm going to play a video of the entire system, so this biohybrid robotic jellyfish in a tank in the lab. And this is a good place to talk about how jellyfish don't have a brain. They don't have a central nervous system and no known pain receptors. So this device is completely harmless. When jellyfish are stressed out, they start to secrete more mucus, and we didn't see any signs of this stress signal. So we're not doing any harm to the animals, and we're very careful to use smaller ends. Of course, large enough for statistical significance, but not enough to create undue cost to the animals themselves. So what is the information that we can actually get using this biohybrid robotic jellyfish? The first thing that we wanted to test was how frequency changes the swimming speed. So to do that, I put the animals in a 1.8 meter tall tank. We had the animals swimming from the top of the tank to the very bottom with this swim controller with 2 electrode design, and it's 2 electrodes for symmetric swimming so that the animal goes straight downwards. We varied the frequency from the frequencies that worked in the past experiments, so between 1/4 hertz and 1 hertz. So from that, we can collect videos like this. So on the top, we have an animal that's stimulated at 1/2 hertz, and on the bottom is the control animal, so it's not being stimulated. And you can see that the animal on the top does swim faster than the animal at the bottom. And I also just want to point out that the LEDs do flash red every time that there's a signal to the animal, but you don't always see that in the camera just because of the frame rate of the videos themselves, especially through webinars. So these are 3 different clips of the experiments. The first is the control at 0 hertz. The second is at 1/2 hertz, and you can see this very powerful power stroke and this strong relaxation as well. But if you go too high of a frequency, then you start to change the kinematics of the animal itself. So here you can see that at 0.88 hertz, the animal kind of curls up, and the muscle is not allowed to fully relax. So you get less subumbellar volume, which results in less thrust and slower swimming speeds overall. So from these videos, we can get plots of enhancement factors on the left, which are a comparison of swimming speeds to the baseline at no external stimulation over the swim controller frequency on the x-axis. So this is one example of a jellyfish, and we can increase its swimming speed up to 3 times when we get to 0.62 hertz. However, another animal of a more oblate shape, so something that's flatter, a smaller aspect ratio, only has moderate enhancements. And there are more examples in between. So we can see this very clear trend where the more prolate or bullet shaped the animal is, the greater the enhancement factors. So the conclusion from this graph is that we can increase jellyfish swimming up to 2.8 times, but the enhancement actually depends on body morphology / the shape of the animal. So again, we can be very clever and choose animals that are more bullet shaped in order to have faster swimming speed. And to see whether this is actually a trend, I also looked at a mechanistic model. So I created a theoretical model that incorporates body kinematics and thrust, drag, and acceleration reaction forces. And you can see the equation here. And the main takeaway from this is that we can calculate the swimming speed to get plots that are similar to what we see from the experiment. So this is a plot of the normalized swimming speeds in body diameters per second over frequency. And you can see that you get a very similar trend in terms of how oblate the animal is and how the body kinematics actually, the body geometry relates to this enhancement factor. And I did this by varying the diameter of the jellyfish and keeping all of the other body geometries constant. But you might be wondering, okay, well, is this just a function of the diameter, or is it actually the aspect ratio? So when we collapse everything to a single aspect ratio, regardless of the diameter, all of the curves collapse onto this one graph. So it isn't a function, it is a function of the aspect ratio and not of the diameter of the animal. So the next question is, okay, we can increase their swimming speeds, but at what cost is this to the animal? And to look at this, I designed a series of experiments to measure the oxygen consumption of jellyfish driven at different frequencies in a sealed tank with a microoptode to measure oxygen. This plot shows that the black dots are individual oxygen measurements, with the mean shown in this dark blue line and the spread in light blue. We can then convert this into plots of oxygen consumption for these various simulation frequencies, and then convert this into that cost of transport plot that I showed in the very beginning. So as a double check here, the reported cost of transport for Aurelia aurita, which is this species of jellyfish, is shown here in blue. And our unstimulated cost of transport aligns very well within this range. I also compared this to one model, which is a power law. So folding in the experimental data from the straight swimming experiments, but not from the option, we can relate power to cubic speed, and these are our expected cost of transport. We can also look at that previous model that I showed, the mechanistic model, that force balance, and these are the expected cost of transports that we would get from those from that theoretical model, which is entirely independent of experimental data. But when we look at the combination of our oxygen measurements as well as our straight swimming speeds, our experimental data is shown here in black, and we only get a 2 times increase in the cost of transport. So I just want to highlight that at this point, we can get up to 3 times an increase in the swimming speed of the jellyfish at only a 2 times increase in the cost of transport. So this suggests that jellyfish potentially exhibit more efficient swimming at faster speeds. In addition to metabolic demands on the animal, we can also consider the power consumption of the microelectronic system. So as shown here, this plot shows the external power per mass over the speed for a variety of different swimming robots in literature. I just want to point out a couple of different robots. So here we have the Jellyfish, which is a purely mechanical robot that is like a jellyfish. We also have a robotic stingray, which is kind of like our medusoid from the beginning of the talk. It is a biological robot. And the Remus 100, which is on an AUV. And compared to all of these different types of robots, our biohybrid robotic jellyfish uses between 10 to 1,000 times less external power per mass compared to these other robots. So not only does this have the potential to probe the limits of biology, but by leveraging animals as a natural scaffold, we can also offset power constraints in robotics. So going back to the spectrum of bioinspired jellyfish constructs, instead of doing a purely mechanical robot and more of a biological robot, we decided to go with a biohybrid robot by integrating microelectronics onto that live animal. And this is advantageous because it's low power and low cost. All of the materials cost about $20 to manufacture per device. It's also adaptable to many environments because these animals are ubiquitous, so we can find them in a variety of these places and deploy biohybrid robots in those areas of the ocean. So then the last part of my talk is about improving robot controllability and working towards the goal of ocean monitoring. So we can look at the robustness and maneuverability of these animals in real environments from those field experiments that I alluded to in the beginning. So last summer, I went to the Marine Biological Laboratory in Woods Hole, Massachusetts with my collaborators. So a big thank you to our scuba divers, Jack Costello, Sean Colin, and James Townsend. They were in the water conducting these experiments for me while I was lazing on the dock. So thank you very much, I appreciate that. And just to refresh on what the lab work looked like, we had the animals swimming straight downward in a large tank. And instead, we wanted to replicate that in the field by having the animals swim in the ocean. So we had a 5.5 meter tall part of the ocean that we were looking at. And in the background of the animal, we had a rope with markers laid at every foot so that we could track where the animals were relative to that rope. We looked at 4 different animals at 0 hertz, so that's the control, as well as 0.5 hertz and 3/4 hertz, just to see if we could see a very similar trend with how frequency affects swimming speeds in the field. And so this is an example of what some of those videos look like. You can see that the jellyfish is swimming freely, it's going upwards. You can see the rope with the markers tracking in the background, and all around the jellyfish are fish and other types of flora and fauna. And you can even see that sometimes the swim controller flashes red, and I've actually captured it on the camera. So from videos like that, we can track the marker centroids. So I'm plotting the red and yellow markers over time. So you have the XY pixels over time. And you can see that the markers track pretty well. We can also track the motion of the swim controller and the jellyfish. And from that, we can get a plot of the vertical displacement over time. So for 25 seconds, the animal travels about 110 centimeters. And so when we collect the data, we can see that when the animal is, or again, the absence of simulation, the animal swims between 1.8 to 3.5 centimeters per second. But when the animal is stimulated at 0.5 hertz or 3/4 hertz, then you can get faster swimming speeds from 3.1 to about 4.9 centimeters per second. So you do see that there are similar trends in terms of the enhancements at externally driven frequencies. So then the question is, how can we incorporate AI to help us in terms of creating these biohybrid robots that we can actually deploy in the ocean? So I just wanted to play this video because you can see that there's a lot of motion from the camera, a lot of motion from the flow in the ocean, the animals moving around, there's lots of fish. You can see that a random other jellyfish is kind of photobombing the camera. And from this, we would really like a way to automate tracking the animal and tracking various features. And we can do that using image segmentation. So this is all future work we haven't incorporated yet because the number of videos that we've taken so far are low enough that I can just run them through MATLAB. But in the future, when we get larger data sets, we're hoping to incorporate things like this image from Google AI, where the computer can segment different parts of the image and show that there's a chair versus a table versus flower pots. And it doesn't matter that there are flowers are all on this table, the computer can still track what this is. So then we'd be able to automate all these different videos when the background is murky or the jellyfish is kind of out of focus or there's a lot in the background. So basically, I just wanted to point out a couple of different features here where in the first image on the top left, you can see that the animal is in very clear sight. You can track the swim controller very well, but there's not a lot of features in the background. And in the other images, we can see that the water is either murky or greener, or the animal and the swim controller blend in with the background. So it's more and more difficult to track those. So going back to our first slides about the goal to use to collect data about different ocean environments, we had a couple of needs for different tools for ocean monitoring. So we want to be able to explore new locations, and have we addressed this? Well, jellyfish, again, are ubiquitous, and they're found in a variety of different environments. So we're hoping that because they're found in these different environments, we can then take advantage of that and deploy these biohybrid robots where these animals are naturally found. Of course, we would have to harden the microelectronic system, but hardening that small system is easier than hardening an entire robot. So we're hoping that that's more feasible. This is also low power, and I hope that I've convinced you that. It's also relatively low cost, again, costing about $20 per device and probably less as we scale up. And we hope that it also minimally disturbs the aquatic life. So as shown from the videos of the animals swimming in the ocean, fish can approach the biohybrid jellyfish robot and they seem to be okay. Of course, we're not actually tracking whether the fish seems stressed, but in the future, when we incorporate sensors on the animal, including small cameras, we can track to see how close other animals get to the jellyfish and whether they actually interact with the animals, and if we can discover new creatures or new types of behaviors that we haven't previously found. So again, we can achieve this with a combination of technological advances as well as experimental work to discover the science. So what is going on with organismal biology and how can we learn that and take advantage of that in terms of creating more engineering tools? We also want to combine that with computational tools, and I'd like to expand that. So it's really nice to be here and talk to all of you guys, and I hope that you guys have lots of questions and can talk to me about the future of this. So I want to talk a little bit about the next steps towards using these robots for ocean monitoring. So in terms of the technological side, we want to incorporate various sensors. Again, this could be temperature or pressure or other things. On the science side, we want more lab experiments to control more complex maneuverability. So all of the experiments I've shown so far are with straight swimming, but we can also make the animal turn if we simulate the animal asymmetrically. So how can we do that in a more controllable way? And what are the swim trajectories that we can actually get in the lab before we move this system into the ocean again? We also want to be able to test various jellyfish species. So the species that we've been working on so far are moon jellyfish, which are the most common jellyfish species, but we can also look at other different jellyfish types to see how broadly applicable this is. We'd also like to continue our field experiments and ocean monitoring, and I just want to take some time to highlight how our field experiments are very preliminary right now, and we're far from the stage where we can just deploy hundreds of these biohybrid robots. So people don't need to be scared about biohybrid jellyfish taking over the world right now. But the hope is that once we incorporate different types of maneuverability and different types of sensors onto the animals, we would then be able to deploy these in larger numbers. and incorporate more not only bio-inspired materials, but biodegradable materials that would be able to break down very quickly. And so it wouldn't matter if an animal like a sea turtle or these jellyfish. In terms of the computational tools, I've discussed how we could potentially use AI for image segmentation and object tracking in a variety of environments as the images on the camera change. And we can also look at CFD to determine optimal sensor placement. So again, where on the animal can we place various sensors like pressure, because we want to figure out what are the forces all around the jellyfish. And not only, we want to figure out areas of the jellyfish that not only experience the highest flow, but also would be minimally disturbed by the animal's own motion. We also want to track the trajectories of the animals to close this feedback loop, which right now is open. So with that, I'd like to acknowledge the members of the DeBerry Lab, Cabrillo Marine Aquarium for providing us with Aurelia aurita animals, the Marine Biological Lab, and our collaborators at the MBL, my funding sources, and I'll leave up my contact information. So feel free to ask me any questions. Thank you very much.